Thanks a lot for the opportunity to speak here. Thanks a lot, Richard, for the kind introduction. Yeah, I must admit that I don't own a Precision NanoSystems nanoassembler. And I've never worked, yet, and I've never worked with a particle that comes out of it. But my last slide will show that that is about to change, and how actually it makes sense that I'm standing here as a keynote speaker. Also, well, facing the challenge is something that we've tried to avoid very long and very successfully over the past years, because there is a challenge, but we've looked away from it for many years, and I will show you some of the work that we've been doing and how successful we were at ignoring the problem, and finally arriving at the issue of how to approach reproducible and scalable nanoparticle manufacture. The story I want to share with you is, first of all, one of the successes that we did, which was not for siRNA delivery, but for small molecules, and that was with polyethylene glycol-coated liposomes, stealth liposomes. 100 nanometers in size, filled with small molecular weight drugs. There we have walked the path from bench to bedside, and I say we, but Bart Metzelaar, who is my colleague, really did the majority of the work in that respect. He works at Enceladus Pharmaceuticals, and he has successfully managed to get a product that was developed in our laboratory bench into the clinic. And these were the images that his invention was based on. On the right, we see his images of rats that received rheumatoid arthritis after adjuvant injection of chicken collagen. And you see in the paws that they develop arthritis, and if you give radio-labeled polyethylene glycol-coated liposomes, they accumulate inside the paws. And on the left-hand image, you see an image from our colleagues in Nijmegen, also from a rheumatoid arthritis patient, where you see where the arrows are indicating that there are liposome accumulation inside the joints that are affected. And what is important to note is the fact that you see the joints in one arm that light up. There we see liposome localization, but the other arm, which is not suffering from rheumatoid arthritis, you don't see any accumulation of liposomes. So they naturally follow or target the joints that are affected. And this was developed later on in Bart's company, Enceladus Pharmaceuticals. And the clever bit basically is, apart from using the polyethylene glycol-coated liposomes, is the fact that he used a membrane-impermeable version of dexamethasone so that it would stably be encapsulated inside the liposome. If you would use a normal steroid, it would rapidly exchange because it can pass over membranes quite easily, so your liposome would circulate, but it would circulate empty after a while. But if you use the phosphate version of dexamethasone, now all of a sudden, because of the charge, it becomes membrane impermeable and travels together with the liposomes to ultimately arrive at the site of the lesion. And that has been being developed now clinically. It's in phase 2 clinical trials together with Sun Pharma, an Indian pharmaceutical firm. And I was working at that time together with him. Well, these are the preclinical data that show his research. Well, first of all, you see a lot of control lines, the empty liposomes, rats injected with PBS, and you see the disease development in this case, where the disease score is marked on the y-axis, and all the control animals show progression of the disease, and with one single injection of the liposomal prednisolone phosphate, you can completely resolve the disease just with a single injection. And that was enough evidence to pursue for clinical development of this project. And I was working alongside with him, and I was working on a project to inhibit angiogenesis. And I was reading the literature, and Judah Folkman is one of the pioneers of this concept. And there was one paper talking about angiostatic steroids. Steroids not only had anti-inflammatory efficacy, but they also could affect the process of angiogenesis. And that would be interesting because we had a pharmaceutical formulation of steroids already on the shelf, so I could begin to test that as well. Well, corticosteroids are often used already in tumor therapy. They are used to reduce the inflammatory responses, antiemetic activity, and cytotoxic activity specifically on hematological tumor cells. But then in the 1980s, and that was again pioneered by Judah Folkman, there are effects on solid tumors, and that might possibly happen through anti-angiogenic effects. But the doses that you need to achieve that actually induce death in the majority of the animals. So you need such high doses that you completely block the immune system, and those animals became very susceptible to opportunistic infections. So it's not really a therapy, but it's nice to show sort of proof of concept that steroids could have activity on solid tumors. But we had our targeted formulation, and at the same time there was also an additional anti-tumor, or the role of inflammation in tumors was becoming more and more prominent, that the immune response inside the tumor is not only fighting the tumor, but may actually in some instances be beneficial. The chronic inflammatory process leads to secretion of growth factors, so it may be the angiostatic effect, but it may also be the immune modulation inside a tumor that may help for these kind of targeted liposomal formulations to affect the inflammatory process inside the tumor. Well, in both cases, you could expect a therapeutic effect. And indeed, that is what we observed in our simple models of tumors that we had available. Subcutaneously implanted C26 and B16 melanoma tumors, where you see on the left-hand side, if we do not treat them, they've grown in a couple of weeks to somewhere between 500 and 1500 cubic millimeters in size. But if you give liposomal prednisolone phosphate, single injection, then you can almost completely inhibit tumor growth at a dose of 20 milligrams per kilogram. And that compares nicely to the free drug, because indeed you don't see any efficacy of the free drug whatsoever. So even at 50 milligrams per kilogram, still you cannot see any therapeutic benefit. So you would indeed need to go probably to these very, very, very high doses that would cause very strong immune inhibition and opportunistic infections, death of the animals, before you would see an effect of the free drug. So liposomal drug seems to work in our simple models of tumor growth. And that was later on developed, and other people took over this research line. This was taken over by Gabriel van der Pluijm at the Leiden University Medical Center. And there they studied prostate cancer metastasis. So they have a bone metastasized prostate cancer model, which is far more predictive, far more advanced than the models that we had with the subcutaneous implantation of the tumor cells. This is a model mimicking the human situation. And there you also see the tremendous efficacy of the liposomal dexamethasone in this case, where we, both in the mandibles and the long bones, you see inhibition of metastasis growth in these animals. And this is also showing this from a bioluminescent data, and also the comparison again with free dexamethasone. And free dexamethasone shows some efficacy in this model, but it's by far not as efficacious as the liposomal formulation. So again, the targeting effect that you get by the liposomal encapsulation leads to local accumulation inside the lesions, and that increases the efficacy inside the tumor. Also in multiple myeloma, we've looked at the accumulation first of all and the therapeutic effect. Again, we try to go beyond the normal subcutaneous implantation models. We try to make a model that is more mimicking the human situation. And in this case, we implanted on the back of the mouse scaffolds, ceramic scaffolds that mimic the bones where these multiple myeloma cells home into. So we're trying to recreate the tumor microenvironment under the skin, and that's partly due to the fact that the microenvironment is really important for the sensitivity of the multiple myeloma cells to treatment. So the microenvironment sort of dictates how it will behave against chemotherapy, but also against dexamethasone therapy. Well, we see a wide variety of distribution. First of all, the usual suspects, of course, liver and spleen show up in the heart region, but also the scaffolds show accumulation of the liposomes. And again, we see a strong inhibition of tumor growth, and again, dose dependently. So the tumor doubling time, which is usually around 5 days, all of a sudden increases to 10 days. And you can also see that again in the bioluminescent image, where you see in all the control groups and also liposomal dexamethasone given in this case at the 1 milligram per kilogram dose, we see an increase in bioluminescence, where it stays approximately the same or perhaps even slightly lower in some animals for the liposomal dexamethasone given at 4 milligrams per kilogram. So that's where we stand now with the development of OncoCort. we performed a full toxicological study that is completed now in rats. Well, we saw no unexpected or additional toxicology well beyond the known effects of exogenous corticosteroid administration, so it doesn't increase the toxicity that we are already aware of in these treatments. The sister product of Oncocort, the one that I was talking about, liposomal prednisolone, that is for rheumatoid arthritis, that is already administered to 65 human subjects by now without any adverse effects. And oppositely, therapeutic effects were observed in rheumatoid arthritis patients. And they were given especially to patients that have a flare-up of disease activity. And in those patients, you see a very strong inhibition of taking off the top of the flare-up of the disease. Well, Calyx, of course, is already registered for treatment of multiple myeloma, so that makes the case for multiple myeloma applications. The most relevant safety issue now is infusion reactions, the hypersensitivity that's also observed in Doxil, but it can be easily dealt with slow administration and pre-treatment with inhibitors. And of course, we have already GMP production. So at least for small molecule weight drugs, with an old-fashioned drug delivery system, we have the ability to make homogeneous particles and make them under GMP conditions. But now for RNA, the situation is very different, and we're not facing it yet. We're only establishing the fact that, that there is a challenge. There's a challenge of reproducible and scalable nanoparticle production for RNA delivery. And we worked on that again in the framework of an anti-angiogenic product, where we were trying to select siRNAs and microRNAs that would be able to inhibit the angiogenic process. And the difficulty, of course, lies in the molecule now all of a sudden. It's not enough, like for small molecular weight drugs, to deliver the drug to the tissue, and then the liposome will fall apart and the drug will be able to distribute over the tissue. But with siRNA, it cannot travel over membranes, so it needs to be brought to the site, the intracellular site of location where it needs to be active. So you need to deliver it all the way up to the final micrometer of its activity, and that makes it hard. It's a large molecule, it's charged, and it has a stability problem because it's sensitive to nuclease degradation. It has a transport problem. It's on its own, it's rapidly excreted renally, and it has an access problem. Even if you would get it to the surface of the cell where it needs to act, it still cannot cross the final membranes because it's too large and too charged. So we went out on a quest to look at to identify anti-angiogenic microRNAs. And we had a library where all 1,120 microRNAs that at the time were identified as human microRNAs were known. And we transfected them into human umbilical vein, primary endothelial cells, and an endothelial cell line, immortalized. And we just looked at proliferation. Well, microRNA scrambled is the control. Then we have a positive control siRNA against polo-like kinase, and our microRNA 7, well, we had several hits, of course. If you do 1,120 microRNAs, there's more than one coming up, but microRNA 7 was identified as one of the anti-angiogenic microRNAs that came out of this screening. And then we validated this microRNA target in a variety of in vitro screens for angiogenesis. So here we looked at the Matrigel tube formation assay, and we see that the number of sprouts is reduced with microRNA 7, and also the cumulative length of the tubes is reduced with microRNA 7. And also in the sprouting assay, where we look at the response of endothelial cells to respond to basic fibroblast growth factor, you see a lot of sprouts in all the control situations. And again, with microRNA 7, sprout length is reduced, and also the number of sprouts is reduced. So microRNA 7 still seems to be a bona fide inhibitor of angiogenesis. Final in vitro assay is looking at the wound closure. So we make a wound inside a monolayer of endothelial cells, that is the upper panel, and then we look how, after a couple of hours, how long or how much of the wound has closed by then, so how the endothelial cells have been able to divide and migrate to close this gap. Well, in all the control situations, you see that the wound is pretty much closed, and again, with the microRNA 7, you are able to largely inhibit this process. But then, of course, for the in vivo part, and then it becomes really challenging. I mean, you can put a lot of microRNA on top of cells in vitro, and we have classical transfection agents that allow you to get the microRNA inside the cells, but in vivo it becomes a lot more difficult. And just for proof of concept, we used electroporation, where we inject the microRNA without any protection, just the naked molecule, and then we put electrodes on either side of the tumor, and then we pulse and it feels a bit like electric fencing. I did it on my finger, and then well, it feels like electric fence. So it's not that harsh a treatment. 10 micrograms was injected intratumorally 6 times every second day, and then you see that indeed microRNA 7 is enabled to inhibit tumor growth. So also in vivo, we see at least an anti-tumor effect, and we examined that in more detail, how that came about, and we see that, first of all, the microvessel density is reduced in the microRNA 7 treated tumors, and we did not observe a direct reduction in the tumor proliferation staining, Ki-67, so that indicates that indeed the predominant mechanism of activity was through the inhibition of angiogenesis. But that was electroporation. It's feasible for distances of about 1 to 2 centimeters, but larger than that, then the electric current that is needed is quite substantially increased up to the barbecue stage where you start smelling it. So that's not really a feasible approach in the clinic. So then we had to go to nanoparticles, and this is the polymer that we designed. It's a follow-up version of the RGD-PEG PEI that we originally worked with. It has a targeting fragment, a cyclic version of the RGD peptide that needs to bind to alpha-V integrins on the endothelial cell surface. It has a polyethylene glycol that is able to shield the particle that is below it. And then we have a polyamide-based backbone to which polycations have been added, all by biodegradable linkers, so that at least it can fall apart instead of the RGD-PEG PEI that was solid as a rock. And if we inject that, and in this time we used an U87 glioblastoma xenograft, then you see that intravenous injections, 3 milligrams per kilogram every second day, we have a similar efficacy to the local injection electroporation protocol. You can actually see that we're doing something with the angiogenesis process because the tumors are pale, they show less vascularization, and also if you look at that under the microscope, you see less CD31, so less endothelial cells. So we are able to make them, but we are able to make them by a very difficult method, or at least it's a simple method. At the same time, quality control is not very high. The only theoretical basis, basically, what we have is the fact that if you add positively charged polymer and you want to have a positive N/P ratio, then it's better to start adding the RNA to the positively charged carrier, because then you will never pass the point where there's charge neutrality, because you stay positive and you keep on being positive. Whereas if you do the opposite, you have a positively charged carrier and then you add that to the RNA, you pass that point where there's neutrality and the particles start to flocculate and you will get large aggregates formed. That's the only theoretical basis basically that we have, because we mix them by having high shear stress, adding the RNA on the vortex to the polymer, and that's basically it. And it was that difficult that at the time when I was working in Washington for a small startup firm, I flew into Tennessee in order to make the particles, fly back, and the next week fly in again, because it was not easily transplantable. And that is what we were stuck with. So, well, we know that the challenge is there, and sometimes you need to fly in and out in order to make the particles and let them be produced with high shear stress, vortexing to the maximum, and then slowly add your RNA. Then you can make the particles, but it's not that easy. How am I doing with time? Do you know, by any chance? 10 minutes. Oh, that's perfectly on time, actually. That's good. Then we went one step further, and I must admit that we made complexity even larger. We did not even care about facing the challenge. It got one step worse, because we started working on biologically produced nanoparticles, the extracellular vesicles. They are interesting from many different aspects. And one of the interests lies in the fact that they're able to transport biological molecules from one cell to another. There are different types. Some of them come from intracellular organelles, the multivesicular bodies. And those vesicles, when they are secreted, are known as exosomes. And other ones bud off from the cell membrane, and they are known as microvesicles. Difficult to discriminate once they are released, because on average they are about 100 nanometers. The exosomes suggested to be a little bit smaller, and the microvesicles perhaps a bit larger, but they overlap, and there are no unique proteins or biomarkers that can distinguish either one of them. So extracellular vesicles is the term that we have to settle upon to describe them. And what is interesting is that they have been described to be able to transport microRNAs, messenger RNAs, from one cell to the next. So they could be sort of nature's way of transporting nucleic acids. So perhaps this could be one of the interesting nanoparticles of the future. At least that was what we thought at the time. And in collaboration with Jacco van Renen of the Hubrecht Institute, we decided to look at that in vivo by using the Cre-LOX system. And it makes use of a Cre-producing cell, so that makes the enzyme that is able to recognize LOXP sites, cleave them out and recombine the two strands. That secretes Cre messenger RNA, not the enzyme as we will see later. It secretes the Cre messenger RNA inside the vesicles. And if that Cre messenger RNA containing vesicle is taken up by a reporter cell that has been designed to have red fluorescent protein production between LOXP sites, but if the Cre messenger RNA arrives, it's transcribed into the enzyme, then the red fluorescent protein is cleaved out and the GFP protein is being made. So if a cell turns from red to green, then Cre messenger RNA must have been successfully delivered. That's the idea, basically, of this reporter assay. Here once again, on panel A shows the donor cell that has Cre and is turquoise, and then you have the non-recombined recipient cell that's red, and then the recipient cell, or the recombined recipient cell turns green if Cre messenger RNA arrives. First of all, we isolate of these reporter cells the vesicles, and they are about 100 nanometers in size. They can be taken up by the acceptor cell, so at least they are internalized, and they do not contain the Cre protein, but they do contain the Cre messenger RNA inside the vesicles. So protein is not present, probably because it has a nuclear localization signal, so it immediately goes back into the nucleus, but the messenger RNA can be recovered. If we use a transwell system where we have the Cre producing cells in the upper well and the reporter cells in the lower well, we can see green cells appearing. So something that passes through the transwell system is able to deliver Cre messenger RNA into the reporter cells and turns green. There's a huge difference in the connection that can be made, apparently, between the cells, because some cell types only show 0.5% recombination, whereas some cell lines show 5%. So there's difference in how well the match is between reporter and acceptor cells. But clearly green cells can be observed. And what's interesting is that controls do not do that. If you inject a recombinant protein straight into the tumor, you don't see any green cells. If you lyse the vesicles before you inject them into the tumor, nothing happens. But if you co-culture the Cre reporter tumor cells, the Cre producing tumor cells with the reporter cells, you see inside the tumor green cells appearing. So also in vivo, this process is maintained. Next to this delivery of Cre messenger RNA, those vesicles contain a lot of other signals that come from these, from the tumor cell, the malignant tumor cell in this case that we used. So we see a lot of microRNAs that are associated with locomotion, metastasis, cell migration. So all this whole payload is also delivered together with the messenger RNA for Cre, and that is something that we investigated. First of all, showing that not only if you mix them locally, you see green cells appearing, so it's a local effect, but also at a distance. So if you have in one flank the Cre producing cell line and in the other flank the reporter cell line, you still see green cells appearing in the reporter cell line in the other flank. So the signals can travel, and they can travel systemically. But what is interesting is that the malignant tumor cells, they move around a lot. That's the, in panel D, those are the cells on the left-hand side. The reporter cell lines, in principle, are quite benign, so they don't move around a lot. That is the red cells, normally. But after they have turned green, so after they have received green messenger RNA, together with all the other signals that come from the malignant tumor site, they start to move around a lot more. So it's transfer of a biological signal over a distance, and it's a complex signal. It's not just a single green messenger RNA, it's a whole lot more that is delivered. But what a pharmaceutical nightmare. This is how it looks like. Many different proteins, many different nucleic acids. How are you going to guarantee that you have the same product, if you think about this from a pharmaceutical perspective, every time that you culture your cells? It's very difficult. The only option I see for these kind of things is that if we would be able to identify crucial proteins that are important for the interaction and then transplant those on a synthetic delivery system, something like that. So we've been very successful in avoiding to face the challenge that is of the reproducible and scalable manufacture. This is about as worse as we can achieve. We've tried that with a number of synthetic systems as well. I'll go over this fastly, sorry, because otherwise I'll run out of time if I have to do this for the synthetic system as well. We've been able to successfully use synthetic systems for the messenger RNA delivery. But finally, we have to face this challenge of how to make these nanoparticles reproducibly. And it was in the framework of this NMBP10 call that we finally came to realize time has come to do something about it. It was started 14th of October 2015. I met Gesine in December here in London, and we discussed this project proposal. And then, well, it seems to be a perfect match, because in the call text it says, formulation of nanomedicines has in general been empirical and often produced amorphous or undefined. And the aim of the research is to achieve excellent quality control of the assembly using self-assembling systems and to have ultimately something that is transplantable from one laboratory to the other. We don't want someone to travel from Washington to Tennessee in order to make the same particles that you were making in Washington. So we have to do something about that, and that's where Precision NanoSystems comes in. This is the consortium that we ultimately designed in December. We got to, so there were 98 proposals submitted in January. 8 were invited in May for the second round. 3 of them were successful, amongst which Be Smart, Brain Specific, Modular, and Active RNA Therapeutics. And this is our bet on how we're going to achieve the transplantability of nanoparticle design. So we put in a machine at every lab in the consortium so that everyone works with the same protocols, the same machine, and we just have to pass around materials instead of that we make the particles and send that from the lab that makes them to the lab that ultimately going to test them in vivo. And well, that's where we stand now. We're about to kick off this consortium the 1st of January, and the first meeting is 3rd of February. So we'll see how, I'll keep you updated on how it performs in an actual setting with 8 partners trying to make the same particle in every country. These are the people involved in the research, and I thank you for your attention. We'll see, we'll see.
We do have time for questions if anyone would like to raise any.
Yeah, sure.
So thanks, that was really good. Question around the exosomes.
Mm-hmm.
So very interesting concept to use them, and I agree that a synthetic approach to make them is going to be the way forward for clinical.
Yeah.
But do you think there's any enzymatics or any processing that happens in the cell that will be required outside of a self-assembly type process?
To help make the exosomes?
Yeah.
Definitely. It's a very regulated process, and the composition can change in response to the trigger by which they are formed. So indeed, it's a very regulated process, and we do not I would say completely, but we do not understand how that response is really regulated. I think there are many reports out there, people reporting, for example, if we look at the uptake side, people reporting fusion of extracellular vesicles with cells, that is the predominant mechanism by which cargo is transferred. Also other people report the conventional endosomal lysosomal pathway, and the majority actually gets destroyed. It's still difficult to interpret what is happening. We are doing a lot of studies, and well, in our hands at least, in the combinations that we use, the micropanocytosis and endosomal lysosomal pathways seem to be predominant. Yeah.
And their creation is, you think it'll make it with the self-assembly process?
I think it's compatible, but then again, we do not. I think in this project we primarily will use biological ones, but then try to take them apart and reload them. And in principle, I think the components are there, but it will be a struggle. I think the best chance for success we have is with the synthetic systems altogether, and that's where this system is actually quite suitable. Yeah.
Thanks a lot for this interesting talk. I have a question to the dexamethasone liposomes at the beginning. You have shown the different organs with this Alexa dye.
Yeah.
And you have seen in this right part where you are interested in high uptake or high accumulation, and you also have seen the heart, liver, spleen. Do you know in these other organs it's due to the blood which is still in the organs, or it's uptaken into the cells? So in like heart, liver, spleen.
In the heart, it's probably still blood that is present. In the liver and spleen, we know that it's primarily macrophage uptake, and actually this is specific for this dye that we use, because that stays inside the macrophages. Whereas the dexamethasone phosphate, after the liposome is destroyed inside the lysosomal compartment of the macrophages, the phosphate is cleaved off, then the dexamethasone is able to travel again, and then it rapidly leaves the macrophages and actually resurfaces in the plasma. So there you see, after 8 hours, you see the dexamethasone peak recurring.
Thanks. This already answered my second question.
Okay.
I'll throw one in,
Okay.
if that's okay.
Yeah.
Also related to those dexamethasone questions, from my years in pharmacy, as a pharmacy student, I know that corticosteroids are not the best drug to give as for chronic diseases, really.
Yep, agreed.
For these liposomes,
Yep.
what do you envision? Do you envision this as a chronic treatment?
Well, at least for the rheumatoid arthritis, it was specifically designed to thwart these flare-ups of the disease that apparently can occur, and in some patients they are corticosteroid insensitive. So you have to go through the complete phase until it sort of resolves, and then it becomes responsive again, and then it's, well, and to take off the height of that peak, there the liposomal corticosteroids come in. Because then it makes sense, because you cannot compete with an oral tablet, with an intravenous injection, or all these kind of things. So it might work better, but it's not warranted against the costs and the difficulty in going to a hospital. So specifically for that indication. For cancer, I think a lot more is allowed, or is potentially possible, because of the seriousness of the disease, the life-threatening character of it. But still then, we would still try to minimize it as much as possible, although the side effect does seem to be a little bit different. Side effect profile seems to be a little bit different than the normal oral corticosteroids, in a sense that we do target the lesions, at least in the experimental animals. We see quite good targeting of the experimental lesions, and that allows you to achieve therapeutic effects at much lower doses. So perhaps we can win something at that end. That will, well, the clinical trials are starting, prostate cancer in Leiden, in Germany, multiple myeloma, so that will be interesting to see how that turns out.
Thank you very much.
Okay, thank you.